you're getting a double whammy tonight. You're really lucky. From two, well, one true Australian and myself, who was a sort of half Australian, and both of us now living here in Maryland in the US, working for the National Institutes of Health. So it's probably worth just saying a word about what goes on here. So the NIH is, it consists of 27 institutes and centres. It actually constitutes about 60% of the world's biomedical research budget. It has a budget of about $31 billion a year, of which we're avidly trying to spend as much as we can all the time. And virtually all of that, at least what we're doing, goes on extramural research, and we're funding investigators in universities and so forth. So about 2 years ago, or a bit more than that, it was recognised that data science was becoming a very important part of biomedical research. And that's what I'm going to focus on. We're going to focus on telling you a bit about tonight in terms of, well, tomorrow morning for you, for what we're actually doing around that. And so I actually report directly to Francis Collins, who's the director of NIH. So I think that sort of speaks to the importance, or at least the perceived importance, of data science in biomedical research these days. And I think it really is we are at a pivotal point of change where, biomedicine has gone from very much a sort of observational and experimental science to very much an analytical science. And that level of change is just accelerating day by day. So I think, and we're very big on bringing computer scientists, computational scientists, and statisticians, applied mathematicians, and many others to try and work on biomedical problems. So part of the reason for my doing this is partly because David's a good guy, but partly because I think that it's really important that you potentially think about careers in this direction. So that's sort of the motivation. So I would say we've come a long way. Just in my lifetime, which of course unfortunately now is quite long. And I just wanted to give you a sense of what that means in terms of the kind of research that goes on. So what you have over there is, David mentioned the Protein Data Bank. It's a resource, it's a database effectively of three-dimensional structures of biological macromolecules, DNA, RNA, protein. What's on the screen right now is the sum total of the holdings of that resource when I started working on this, actually when I was a graduate student at Flinders University of South Australia. And what used to happen is that the data resource itself was at Brookhaven National Lab in New York. And these tapes used to come by sea mail every 3 months. It used to take about 3 months to get there. And then I would avidly essentially print out these resources, the details of these proteins, and look at them by hand. So I know this is sort of, to a young audience like you, it's sort of, you're probably already glazing over, the old man's going, boring us to death. But I think it's quite remarkable that now I can do the same thing. And here's an example of a visualization of the complete Ebola virus. It's the proteome of the complete Ebola virus. We happen to be working on targeting VP24 as a potential target to treat Ebola patients. But so this is essentially analyzing all of that data and visualizing it, all obviously happening in effectively in milliseconds relative to what happened that used to take 3 months on a very much smaller amount of data. So those 20-odd structures that you see on that sheet, there's now over 100,000 of those, and it's growing very rapidly. So I think it's a reflection of what's changing. The other effect, which is sort of it's worth pointing out, is that we've been very successful. If I was giving you this lecture in 1900, I'd have already been dead about 15 years. The life expectancy has gone up dramatically. Vivian's looking at me. Has gone up dramatically in that time frame. Even in my lifetime, it's gone up significantly. So I think obviously that's not all due to biomedical research. There's a lot of other environmental and other factors. But it's quite a remarkable set of circumstances. And I think it's in part tied to changes that we've seen you can tie some elements of this to what happens has happened in computing and computing capability. So there's some level of correlation here. And if you start looking at things, so first of all, it started with sequencing and the computational analysis of sequences. I think protein folding is another example where computing has had great insights into helping us understand how proteins fold and what the implications are for disease and so on, particularly around misfolded proteins. And then more recently still, the emergence of using this computational power to look at and factually simulate whole cells. The thesis defense I've just come from remotely was doing exactly that. It was looking at the complete protein complement of a fairly simple cellular system and how that was being synthesized and essentially simulating that kind of activity. And then I think there's search around what we do with databases and just vast amounts of online information that really all have contributed to our knowledge and our understanding of biomedical systems and how to address this with respect to healthcare. And I'm just going to give you one or two examples of how of what the impact is, and then sort of how we're trying to further exploit these possibilities that are brought about by the very fast or decreasing costs of computation. You can look at it in different ways. This happens. I was actually at the San Diego Supercomputer Center involved with it at least for many years. And this is just a timeline of some of the activities that happened around those kinds of technologies, which turned out to be very important, including developing algorithms to compare protein structures, which when we originally did that, that was not possible to do that on anything but a large supercomputer to at least compute pairwise comparisons of everything that existed at the time. So these kinds of resources have been absolutely critical to advances in biomedical computing. But I think I should say that there's also words of caution in all of this, and I think it's really important to think carefully about the problems that you address with computing. It's not just a matter of throwing cycles at a problem. It's really thinking through what the implications are. An example of that would be there was a lot of activity. In fact, it was a so-called, very high-profile project at one point to actually to use the computer to ab initio fold a protein. So, in other words, to take a string of amino acids, the building blocks of proteins, and actually determine what the three-dimensional structure was ab initio. This is, and still remains to some degree, one of the sort of unsolved challenges. It's a grand challenge of molecular biology. The relevance to this discussion is that for all the effort that went into that and all the compute cycles that were thrown at it, in the end, where the greatest advances were made was not from raw computation, but it was actually a growing body of experimental data that could be used to simulate those structures through a process called, among other things, homology modeling. So while all that compute was thrown at it, it was really the availability of far more data that made a big difference. And you can make the same kind of analogies. Another grand challenge many years ago was the idea of weather prediction. Well, of course, a lot of that, at least in the short term, has been replaced by just looking at data of satellite imagery. So I think there's really understanding the problem and where the data is today and where the data will be in the future to define how you think about these problems. But, even though we're living longer and, there's still some fairly serious kinds of problems that we're concerned about. And I'm in the inner sort of sanctum of the National Institutes of Health. So we talk quite a bit about our relationship with Congress and the US government. And, you know, there's some unrest at $31 billion. We're still in a situation where we have really too few drugs for the amount of money that's spent. It takes way too long to get them to market. And, you know, even today, and I'll say a bit more about this in a minute, they're not personalized. So in other words, everybody effectively takes the same drug in the same amounts. Obviously, it varies a bit according to body mass and so forth. But it's not, this is not very personalized, and it's not very effective. We ignore things like rare diseases because there's not a lot of money in them. And yet, for those who are afflicted or families of those who are afflicted, it's a severe problem. We run clinical trials on small numbers of patients. And they're incredibly expensive, and they're not very retroactive. So, you know, the idea of using existing data on patients is very attractive. Patient-centered outcomes, as it's referred to. And that involves using a lot of upcoming data, which I'll say a bit more about as well. In terms of education and training, I'm sure you're doing better there, but the situation here right now is, you know, in big data and data science and so on, we have a real shortage of workforce who can, you know, address these analytical problems. And then a big issue here, and perhaps in Australia too, is that research itself is not particularly cost effective. There's a lot of concern here about the inability to replicate important studies, partly because data is not available or software associated with the study and so forth is not available. And there's also concern about, you know, someone does a piece of research, the moment they decide that they want to publish that, it can be up to a year before that work actually appears. If you're afflicted with a disease that means that you're likely to be dead in 3 to 5 years, a year is an awful long time. So, you know, these are all issues that we're trying to grapple with in this sort of general way. At the same time, there are other kinds of you know, I would say ferment in the system, which really intrigues me. And this is just to remind me to tell you about a little story about one such sort of situation, is that, and Dave mentioned I was editor-in-chief, or still am at some degree, of a journal called PLOS Computational Biology. Well, I actually got a paper several years ago now on pandemic modeling. So it was actually modeling what happens during a flu pandemic. And in other words, to try and predict and essentially treat the condition in a better way. I looked at this paper. It was actually, it was quite an unusual technique, and it had some really interesting outcomes. And it was written by one author. And I was pretty intrigued by this. Normally, I would have just sent it directly into the journal and not looked at it myself, but I was intrigued. I looked at it. I sent it to a guy called Simon Levine, who's a Kyoto Prize winner who works in this area at Princeton. He said, yeah, this is really something quite special. So because the single author lived in San Diego, where I was at the time, I decided that I would meet with her and discuss the paper. I was so impressed when she came to visit that I arranged for her to give talks in San Diego and in Princeton. So what's the big deal about all this? Well, the big deal was when she actually wrote this paper, which was reviewed by Science, she was 15 years old and she was a senior at La Jolla High School. So she wrote a first-rate piece of science at a very young age. I tell my daughter this, when I tell my daughter hears this story, she hates it because she was about the same age at the time. And clearly that this was an exception rather than the rule. But it tells you that, you know, under the right conditions, anyone can contribute. And the reason she was able to do this is because she actually wrote to Wolfram, got a free copy of Mathematica. She wrote to the San Diego supercomputer and got center and got many thousand hours of computing time to run these simulations. And she had access to the open access literature. So she was actually able to make a pretty significant contribution. So these are all things that we try to take into account in what we're doing at the NIH. So, for example, we recently had a workshop with gamers because we felt that gamers could really contribute skills to biomedical problems. And it was actually really successful. And that's led to a number of ongoing activities. So so this is part of the ferment. Another aspect of all of this, of course, is just the amount of data that's coming on each individual patient. I'm going to tell you a little about the US project, but this is the 100,000 genomes project that's going on in the UK, where they're sequencing all of the genome of that many people, focusing on those that have particular conditions to get a much better genomic footprint of a series of different diseases. So this is, generating a large amount of information. In our own case, and I was involved with this in the not long. I've only been at the NIH about a year and a half, and I'd only been here about a month or two when Francis Collins, the director, came to me and said the president wants a brief on the intersection of big data, genomics, and personalized medicine. And so we all avidly ran away and wrote this brief. And it was really cool to write something that you thought that you realised the president was going to read. He'd already put forward a bill around personalised, which is now called precision medicine, when he was a senator in Illinois, and this ultimately led to this announcement in January of the U.S. government putting $215 million into some initial studies to look at this so-called precision medicine. And what that amounts to, and it's about to start, is developing a cohort of over 1 million volunteers where there will be extensive information ranging from genotype to phenotype. So their genomic information, everything associated with their patient record, and also information that's collected possibly by the patients themselves through mobile devices and so on, so that you begin to build up a much more detailed and ongoing so-called longitudinal study of these individuals, including things like their lifestyle, biological samples and so on, where you can really start to look at this and look at people on an individual basis. So what value does this have? Well, I'm going to give you a sort of example of that in a second. Let me come back to that. So here's an example of that. So what this is a so-called comorbidity network. And what it really, each node on that network is a condition. So for example, there's a node, I don't know if you can see my mouse, but there's a node here, COPD, and the thickness of the edge to respiratory failure is essentially the likelihood, based on the whole Danish population over a period of 15 years, of you going from one condition to another. So this is just mining that kind of healthcare information on individuals that I mentioned before. And suddenly, this is a cursory view of this, but it's really becomes an enormously powerful predictive tool about the likelihood of going from, in this case, from one condition to another. And of course, you can provide intervention into this process to actually facilitate. So you're moving towards preventive medicine. So this is I think a big data type problem with this kind of data. And in fact, just to indicate the amount of data that's sort of coming, this was the National Library of Medicine has a resource called the National Center of Biotechnology Information, which some of you may have looked at it, used. It has a whole series of databases. It has a resource called PubMed and PubMed Central, which is the literature. These total contents of that national and international resource, at one point, whenever it was, 93, I think, fitted on one CD-ROM. There's now essentially 40 petabytes of information. And so, it's just an indication of where this is going. It's estimated in the kind of data I just showed you that for an individual, their longitudinal, their lifetime study, their genomic and phenotypic information will probably be the order of estimated about 40 terabytes. So 40 terabytes times 1 million patients, which is actually a small sample. You're really getting into this sort of big data stuff. And we're addressing this through a program which is called the Big Data to Knowledge Initiative at the NIH, where we're trying to leverage these kinds of developments and possibilities with a series of activities, which I'll say a little about, and then I'll get Vivian to say something about as well. She's waking up. Oh, wake up. So how we're doing that is we're trying to sort of put all this together as a sort of what we call a digital enterprise. So we're trying to establish an ecosystem around all this data, software, narrative, to actually effectively accelerate the process that leads to these improvements in healthcare. And I'm just going to say just a little bit how we're doing that, and then I'll get Vivian to say. Vivian's the techno geek in the group, so she'll describe a little about some of the things we're doing, particularly in the cloud. But we're kind of looking at this, what we're doing, from sort of three perspectives: community, infrastructure, and policy. And all of that is based on the idea that what people really care about is getting their research done. So they don't really care so much about the policies and to some extent the infrastructure. They just want to get the research done. But so that's what the drivers are. But underpinning that are these kind of activities around these three major areas. So one of them is, and as David mentioned, I'm big on the idea of open science and data sharing. So the US government, and I think it's certainly prevalent in Europe as well, and I'm not quite sure on the situation in Australia, but on data sharing. So the US government has essentially stated that all data that's generated by the US government on taxpayers' money is to be made, obviously, unless it relates to national security or the individual rights of a patient, will in fact be made public. So we've been developing various plans for doing that, and I won't go into the details of that, but what it really implies is that there's now regulation that much more data will be and continue to be made available. And in fact, I won't go into details of what we're doing around that, except to say that investigators will not be getting new grants over time unless they make the data that they've already generated available. And we're working on ways of actually checking that kind of thing automatically. So it really pushes folks more towards data sharing. And the hardest thing to change of all, of course, is the culture. There's not a particularly strong culture of data sharing. There is in certain areas of biomedicine, particularly genomics, but not so much in other areas. And we're, changing that culture is the hardest thing of all, but we're sort of pushing on that as best we can. One way of achieving that is the idea that the way we measure your success as a scholar right now is totally broken, in my view. So you're measured by how many papers you produce. Maybe it's a little different in computer science, but certainly in biomedicine, in supposed high-impact journals. Well, that sort of, in some senses, it's okay to a point, but it doesn't really reflect the true value of different types of scholarship. An example of that would be from my own situation. I have a paper that's been cited now about 30,000 times. It's one of the most cited papers in all of biomedicine. But the interesting thing about that paper is no one has ever read that paper because it's a paper about a database. We have no way of citing data, so we have to write a paper about the data so we can have something to cite and get credit for it. It just doesn't make any sense. So we're moving slowly towards a situation where data citation itself it becomes important. So, a well-formed data set is that other people use and do research from is important, and you should be rewarded for doing that. So that's an important aspect of this. So those are just examples of policies. And I'm just going to say a little about the sort of how we're structuring this, and I'm going to hand it over to Vivian to tell a bit about what we're doing in the cloud. Come over. So we're funding a whole series of centres around the country that are working on different types of data. Some of them are doing all of everything from genotype to phenotype. Some are looking at one or two, for example, looking at mobility data. For example, a lot of imaging data around people with spinal injuries and things like that, and how their mobility returns and what the implications are, that sort of thing. Using social media data and the implications of that for healthcare, for example, Google Flu and these kinds of things. So we're funding a whole series of these centres, and then they produce data, software, narrative. And what we're trying to do is to build this ecosystem. We're essentially funding some other activities to pull it all together. So the DDICC is actually a data discovery index consortium that all of the content that they produce, the so-called research objects that they produce, the intent is that they will be indexed, and that index will then be made accessible. So you can actually then find, for example, data. So we work under what's called the FAIR principles, which is to be able to find, access, which means effectively use, interoperate, and reuse this content. And that's not trivial to do today. You can spend, and you probably have these experiences yourself, you spend an inordinate amount of time just finding the right piece of software, the right data to use to begin to address a problem. So we're trying to, through these software indexes, through standards indexes, to actually sort of facilitate that process. And we're doing that, including other labs, in something that we call the Commons. And I'm going to let Vivian say, you have to pick up. This is, tell you a little about the underlying architecture and things that we're doing with that. So over to you. So I put you on a spot.
I haven't seen these slides, so I'm trying to guess what he's going to get at here. So we're going to talk about the Commons. I'm going to look at what your next slide is. Oh, this is my stuff. Thank you.
Your slides, don't worry.
All right, so this is an old slide now, but what we're looking at, Phil was talking about all of the things that go into making data interoperable. So what our job is to now figure out how to do this in a technical sense as well. And so the Commons is all the principles that Phil has just talked about, so you can make data accessible, etc. But we're talking about it in three pieces here. One is the computing platform, because you're going to need one, particularly for the large data sets that we have. The other one is the ability to search when you've got data on this actual platform, and you have ways of handling the digital objects in there with UIDs. Now, this is an old slide, and the intent here is discoverability and interoperability of objects. And I'm going to have to figure out what the next slide is to be able to tell you something intelligent about it. Oh, I'm going to tell you about the computing platform next. Okay, this is the Commons framework that I was putting together. Wow, this really is an old set of slides. We can send you updated slides if you have interest in these. The conceptual framework uses a computing platform, and we use, we're looking at three, particularly the public cloud platforms. And here we're using Google and AWS, which are the usual suspects here, but Microsoft is also coming up the rear, so to speak, with their Azure platform. They were kind of out of the loop for a little while, now they're back in. Primarily, these public cloud platforms are being used by NIH researchers because they have very large data sets, and essentially what they're doing is they move them to the cloud, they run whatever analysis they're going to do it, and they bring it down. Now, this is true for mostly folks that have computational backgrounds, bioinformatics backgrounds. It is still not that easily available to biologists. I think all you guys are computational side folks, is that right? Right. Well, I think there's plenty of biologists that still run from the room with their hair on fire when they see a command line. So a lot of the things here are really aimed at high performance type computer folks. But what we're seeing is an increasing number of NIH folks who use these platforms. And the reason for that is so that they can actually do the computes they want to. And as I said, the usual subjects are Google and AWS. But we're also seeing other academic cloud platforms taking up. They don't have the scale that Amazon and Google have, but they have content knowledge, and they know better how to handle some of the data and actually organize it from both computational services within those systems and actually how to represent that data and make it useful to biologists. The other thing that we're doing is working with supercomputing platforms, and part of that reason is they've been, certainly for the supercompute facilities available in the United States, there's quite a large number of supercompute facilities in the United States. I think there's like 10 or 12. they're primarily funded by the Department of Energy, DOE, and the National Science Foundation, not NIH. And so NIH PIs don't generally have a lot of access to that. And essentially what they do, though, is allow you to manipulate and analyze very large data sets. They have a lot of staff that are present, available to them, that have both the computing IT infrastructure knowledge as well as the bioinformatics knowledge to set up the analysis that you need. And so essentially what we're doing is working with a variety of these groups around the country. So for example, there's the San Diego Supercompute Facility, which Phil was associated with. There's the Oak Ridge Laboratories down in Tennessee. There's TAC, which is the group down in Texas. There's a collection, and there's Pittsburgh Supercompute Facility, who've all been talking to us and all doing a whole lot of work in biocomputing. I've done a lot of work with NERSC, right, which is looking at the biocomputing in Berkeley. We're also looking at other platforms and how we would do this in-house. So there's a lot of work being done in that area. The other thing we're trying to do here is look at how we would handle research objects and APIs in terms of making things interoperable for the actual framework that we're talking about here for the commons. And so what we're looking at is how we have, once you have a platform that you can use, and as Phil was saying before, you've got research objects you've got to find, and it's really what you need is metadata associated with those and IDs, unique IDs for that. Although that's starting to happen in biological sciences, it's not always there. And so I think what we're trying to do is to help do that over a long period of time. We're also wanting to move large data sets like the model organism databases, which are commonly used by many scientists, and other large public data sets into these environments simply so people can use them. As Phil was saying before, search, you also want the ability to search on these platforms and be able to discover the data. And the last two points here, actually, the second last point is the APIs which are being developed. There's the Global Alliance. GA4GH is a global alliance of genomic data, health data. It's primarily genomics. They've been working with pharma, biotech, and academics here in the United States and in Europe to develop a series of APIs to be able to question the genomic data, and reusing some of that in that environment. It's very useful. Okay, what am I? Oh, there's me playing guitar. Right, okay. So the next steps we've been doing, thank you, Phil. That's what I do part-time. The next steps are really, we've been identifying projects for some time, and we've actually got a whole stack of them. As I said, these are old slides, but if people are interested, we can give you a much more updated version of the framework of the commons, and also give you an idea of some of these pilot projects that we're actually identifying to use the cloud, developing APIs and other web services to query this information, developing stacks across this that look at... you got a slide on that? Okay. Yep, here's the stack slide here I was looking at. Here's some of the stacks that we're looking at developing. So this is the stack we're looking at the moment, which is the compute cores, which I was talking about before, which they could be HPC or cloud, or you can run HPC on cloud. You need some sort of software that you can basically look at indexing, clustering, searching. I've just used Hadoop here as an example, but there are plenty of other ones like the Apache Lucene systems that are coming up. We're seeing a lot of those here. Then you actually have a layer here of biomedical data where you actually want to search that data, and you want to have software to do analysis for it. It's not necessarily a web service. It's actually the analytics of the data you're doing. And you also want the APIs to be able to operate on that environment, obviously. We've also been exploring the idea of an app store or a digital marketplace so that biomedical scientists can actually utilize this in a much easier fashion. I think that's the end of what I'm going to say. I think we're going back to Phil.
Okay, thanks, Vivian. Good job, but completely out of surprise. So just to say something about where, if there's any interest from folks there about engaging with any of this kind of work, you know, we'd be more than happy to hear from you, because we have a number of sort of training and opportunities, both for graduate students and postdocs, and I'd really like to see some international collaborations around this. So if you have an interest, let me know. So, we're going to be over there in November at some point. You know, we're essentially doing training and offering these kind of opportunities either for people who want to be trained to be really biomedical data scientists, which is the top right-hand side, or people, which perhaps is not relevant to this audience so much, but people who are really experimentalists who want to begin to start working in data science more. And then, you know, we're also developing a whole set of fostering a whole set of online courses and other activities that can be used remotely. That's the bottom left. And then we're also doing things internally to NIH because, you know, there's this change to really much more of an analytical science. I mean, I think there's a lot of retraining that needs to go on, even in somewhere that has a rich research environment like the NIH. There's a whole coordination center around that. So these are sort of a set of problems that I've stated in general terms for the kinds of things that we're looking to have people do research on. So if you have an interest in this kind of, and these are not cast in terms of the biomedical problem, but really the underlying data problems associated with that. So we have a lot of issues about homogenization of disparate, large, unstructured data sets. And that includes things like the electronic health record. So a patient's health record might have some level of structure to it, and it might have some form of ontology and controlled vocabulary and so on. But so much of that record is, in fact, purely unstructured information, very often textual. And how you can homogenize across multiple patients in different ways to get meaningful outcomes from that, as an example. So generalization of that is really deriving structure from unstructured data. A lot of work obviously goes on around image analysis. And so the ability to feature map from and compare image data. A lot of MRI data, for example. Clearly lots of work to be done there. And, you know, multi-dimensional data sets of various types, particularly phenotypic data. I think there's a lot of room for improvement there. Causal modeling of large-scale dynamic networks and discovering from those networks. Again, much more work needs to be done there. And then, of course, a lot of the data that we deal with is actually quite sparse and certainly irregularly sampled and noisy. So these are where we're looking for improvements. And we have problems, but we don't necessarily have the solutions or the ability to, the computational skills for those solutions. So that's kind of what we're looking for. The outcomes that you might see from that, and just some of the things we're currently working on, is the idea of improved outcomes from surgeries with children with cerebral palsy and gait pathology by analyzing mobility data, e-health data from mobile devices. That's being applied to using sort of mobile devices to record smoking habits and weight and dietary habits, with the idea of prompting people to improve those habits. The personalization of cancer treatment. I mean, we're fast coming to the realization that cancer is not a disease. It's not even a complex disease of multiple aspects. It's that everybody, each individual essentially has a unique cancer when they're unfortunate enough to have it. And the idea that we personalize the treatments more and more is an example. And I can go on and on with these. Mental health and addiction are two other examples where we're really looking to see improved outcomes. And computation and analytics are going to be key to that. So I'll just summarize that by relating a story that I heard several years ago from this guy, Josh Summers. His situation was, and it really highlights, I think, what we need to do. So he was a freshman at Duke University in engineering, and he suddenly started getting these dizzy spells, and he went and had an MRI. And after his MRI, he was 22 or 23 years old. A doctor came in and essentially said, I'm sorry to say that you have Chordoma disease. And he had no idea what that was. He tried to look it up. Without access to the literature, which he didn't have because so much of the literature was closed, he got a very cursory view of the nature of this disease, but it's a form of brain cancer. When he went back to his university and get online and into the journals, he realized that his prognosis was probably the order of 5 to 7 years. So he could have gone off and started partying, but he didn't do that. He decided to go and work on the disease himself. And it turns out, quite fortuitously, that the only US-funded researcher in the US was also at Duke University across the campus in the biochemistry department. So he went off to work with this guy and was absolutely aghast at what he found. He found that even people in the same lab were not sharing data, that there was really an incredible lag time between when data was produced and when it was made available, when knowledge was made available. And it relates to what I said earlier, is when you only have, and here's a living example of it, when you only have 5 to 7 years to live, a year to get a publication and then not release the data just seems to be totally unconscionable. And we are making progress. I mentioned that we're making progress, and the lifespan of individuals through the kind of work that we're describing here, but in many other aspects as well, of course. In the developed world, we've seen enormous improvements. But on the other hand, if you live in Angola, your current lifespan is about 40 years. So we have much, much work to do on a global scale. And I'm hoping that you will all be inspired to work on it in some level. So thanks very much for listening. We have a good group of people that work on this at the NIH. I won't go into naming any of them who they are. The guy at the top, the good-looking guy on the right, is myself. The other guy is Francis Collins, who's the director of NIH. I had to be sworn in. It's really scary. But there you go. And I encourage you to reach out to us. I didn't put Vivian's email on here, but I will forward any email you send directly to her.